research students of PhD, MTech, and MS programs, out of which 18 students already received their PhD degree. So today he will be speaking on the topic current from living systems, inspiration from nature to generate power and develop sensors. I will now pass over the stage to Gusami sir. Over to you, sir.
Hi, good afternoon to you all. Am I audible?
Yes, sir.
Yes,
Okay.
sir.
So, first of all, I am thankful to Honorable Chancellor of Assam Downtown University, Dr. N. Datta, then Honorable Vice Chancellor, Dr. N. Sitalukdar, then Dean, Engineering and Technology and Research, Dr. Sunandan Borua, and Advisor of Research, Dr. B.G. Uni, and all the community, all the members of Assam Down reputed University, Assam Downtown University, for inviting me to deliver this lecture. So, the topic which I have picked up today is a purely interdisciplinary subject. It involves chemistry, physics, material engineering, design, computer science, mathematics, and of course, biology is the kingpin of this subject. So, considering the diverse background of the audience today, as I am informed, so I have prepared this lecture, a general lecture. However, if any query arises, if you are interested to know detail on some of the technical aspects, please don't hesitate to intervene me at any point during my lecture. So, the topic is the current from living system, inspiration from nature to generate power and develop sensor. So, there is two emerging technology for the next generation healthcare and allied field as widely opined. These are one is biosensor, which is mainly used for sensing applications, as we know well. One of the good example is glucose sensors, which are available in our local pharmacy and local market itself. The other device is biofuel cell. That is usually a power generating device, but that can also be used as for sensor applications. So this biofuel cell, it produces current, but the level of power is not comparable to the chemical fuel cell. So power generation is low, but it has certain other advantages which do not have in chemical fuel cell. So that I'll be describing in course of time. So let me first describe briefly one by one what is biosensors and what is biofuel cell. Then I'll be describing their applications, current and future applications, and their markets. Afterwards, I'll be discussing our main topic, the source of electrons or the current for these systems, and how we are inspired to develop these systems from the nature. Then at the end, I'll be briefly highlight few of the work which is going on in my laboratory. Now coming to the biosensor, biosensor is basically, it is an analytical tool. It is small, so that's why it is portable. It is a simple device, so it can be used by semi-skilled operator, or if it is used in clinical sectors, then patients themselves can use it, or a layman can use these sensors, as because it does not have any technical nitty-gritties, simple operation. This analytical tool should be reliable, and overall it should be inexpensive, so that it should have a market and should be able to use by the common people. Now, you might wonder that there is sensor in the nature itself. We see the natural sensors. One good example which is surrounding us is, you might have seen this plant. In our local language, it is called Nilazibon, touch me not. Scientific name is Mimosa pudica. If you touch this leaf, immediately whole bunch of leaves collapsed. So it is a good example of pressure sensor. We need not have to go to the plant kingdom. Sensors are with us itself. We have Pancha Indriya. We can smell, we can see, we can sense, we can hear, we can taste. So Granena, Drishti, Shravana, Sparsena, and all these five sensors are already with us. It is not a new thing. These sensors are omnipresent. It is not only in plant and big plant and animal system. It is even in coronavirus itself. As we know, this coronavirus, it cannot enter our body in our cell if it does not get the receptor on the cell surface. Its receptor is called a C2. So its spikes protein must locate the receptor which is present on the cell surface. Then only this virus can dock on the cell surface and deliver the genetic material to the cell for its reproduction and multiplication. So these sensors, natural sensors, are omnipresent. But these natural sensors are amazing, where they perform their jobs. But these natural sensors, they are evolving through thousands of years, particularly for their growth and survival. But the analytical tool which we would like to design is called biosensor. It is difficult to use these natural sensors directly, but this kingpin of these natural sensors, which are the biorecognition systems, if you can utilize this biorecognition system by extracting from the natural sensors, we can expect a very specific kind of biosensors, and that is the biosensors. So biosensors should have some certain characteristics. It should be selective. It should have fast response. Response should be within seconds. It should be sensitive. It should be able to detect in sub-millimolar range. But again, it depends what is the target analyte. If target analyte, for example, if you want to detect hormones and those kind of things which are present in a very, very low quantity, then you may have to go to the picomolar, femtomolar level. But otherwise, if you want to detect, say, sugars, glucose, or all those general analytes, then you need not have to go to that level. You may have to go to the millimolar level or micromolar level. It should be accurate. Whatever the results you are getting by using these sensors should be accurate, it should be reproducible. If you use these sensors again and again, you will get the same results again and again. It should be independent of physical parameters. This is one of the very critical issues. Normally, whatever we do in a laboratory, we generally rely on adjusting the pH, adjusting the temperature when it's tiring. So those kind of physical activities should not be involved in operating the biosensors. It should have short recovery time. Once you use the sensor, then the sensor should be ready to analyze the next sample in a short span of time. And importantly, it should be functionally stable. Both operation as well as storage stability, it is a very important thing. Otherwise, keeping these things in the market, pharmacy, chemical store will be difficult for a long time. So if you see all these parameters, it is a very suitable system for decentralized testing facility, point of care applications, home diagnosis, and on-site applications. So as because it is portable, it can be used for remote location applications in resource limited environment, where resource is there, current is there, skilled manpower is there, their centralized laboratory can be set up. But in most cases, particularly in underdeveloped and developing countries, we have a limited resource. So this kind of analytical device is more useful because it does not require additional resource. Now, as I have already mentioned, the two things, one is biosensors, one is biofuel cell. Now, as far as the biosensor is concerned for the present talk, I'll be talking mostly on electrochemical biosensors because these biosensors involves the generation of the current, which will be considered as a signal. So this is a general configuration of electrochemical biosensors. Here, if you see, one of the important thing is that this biological sensing element. This biological sensing element, as I have already mentioned, that we are trying to extract it from natural systems. It may be an enzyme, microorganisms, it may be DNA, it may be RNA, it may be even cell membrane also, or it may be a whole tissue also. So this is the biorecognition element. And once this analyte will interact with the biorecognition element, then these interactions will lead to some kind of signal. And in electrochemical biosensors, this signal is transduced to electrical form on the surface of electrode. So electrode is mainly used here as a transducer. So as a result of that, the current is generated in the system. So one typical electrochemical biosensors, which is rely on a Faraday current, is this is the picture I am showing here. So basically, it has a 3-electrode system. One is called working electrode, another is called auxiliary electrode, another is called a reference electrode. So in a working electrode, like here, you are dropping your blood sample or whatever the sample, then immediately you will get the results. So this kind of sensors are basically glucose sensors, which are available in the local market or in the global market, are of this kind of things. So now, why this sensor? Because these sensors are very fast. You will get the results very quickly. It is a highly sensitive. It is obviously selective, as because you are using a biorecognition element, which confer the selectivity to the system. It is a label-free system, as because unlike the optical sensors, where in most cases you need to couple a label like fluorophore label, here no labels are required. And interestingly, here sample, you need a very small quantity of sample in the microliter volume, or even without wasting the sample also you can do it if it can be implanted on the body itself. And obviously it is a very small size. So now coming to the biofuel cell. What is biofuel cell? Now before describing the biofuel cell, now I am coming to the back side of the word cell. Cell, I think we know well. Cells we are using in different systems. Nowadays, each of us has the cells in our mobile phone. So these cells, basically, whether it is a biofuel cell or cell, these are all electrochemical devices because it involves electrons. And the reaction on the electrode is involved. But in cell, what is happening? Here, the chemical energy is stored in the system. And so long that chemical energy is there in active form, that battery will work. And once that chemical energy is consumed, the battery, we call it, is a damaged, it does not produce power. So as because this chemical energy is stored inside the system, it is a thermodynamically closed system. Now coming to the fuel cell, then I will come to the biofuel cell. So fuel cell, it is also an electrochemical device that convert fuel from external source to electrical current. So as because it utilizes the fuel from external source, so it is a thermodynamically open system. So this is the basic difference between fuel cell and our normal cell. In a normal cell, the fuel is inside, it is stored, so it is a thermodynamically closed system. And in a fuel cell, we are supplying the fuel externally, so it is a thermodynamically open system. So it is a general configuration of a chemical fuel cell I am displaying over here, where hydrogen is used as a fuel. So in this fuel cell, 2 electrodes are there. Both the electrodes act as catalyst. One is called anode, where the fuel is oxidized, and other is called cathode, where oxygen, primarily oxygen, is used as a reductant, so oxygen is reduced. So main working principle over here is that when, say for example, here hydrogen we are using as a fuel, so hydrogen is oxidized over here, protons are generated, electrons are generated as a result of that split up of hydrogen. So the electron moves to the outer circuit, proton moves to the internal electrolyte to the cathodic compartment. And in a cathode, the catalyst which is there, it reduces the oxygen by taking the proton and electron from outside the circuit and ultimately generate the water. So it is a very clean technology. No emission is there. So this technology, actually, it is not a new thing. It has been, first principle was reported in 1838. Now, the biofuel cell is basically a different class of this chemical fuel cell. Now, what is biofuel cell? In a biofuel cell, these components are different from the chemical fuel cell. In a biofuel cell, the catalysts which are used on the electrode, these are mostly biological catalysts. This may be enzyme, microorganism, etc. Fuel which is used over here, it is a broad range of fuel. You can use many renewable carbon substrates like carbohydrate, alcohols. Even you can use non-renewable carbon also, non-renewable fuel also. If your that enzyme or microorganism which you are using on the electrode as catalyst will oxidize those compounds. Operating temperature is generally 20 to 40 degrees centigrade, normally room temperature, but it may go beyond that also. It depends on if your catalyst is stable, for example, 80 degrees centigrade. If your enzyme is stable, it will be able to work at 80 degrees centigrade also. But it is always preferred to operate the system in a room temperature so that you need not have to waste external energy to heat it up. So biofuel cells are working particularly in a physiological pH, room temperature. And if you see all these characteristics, the biofuel cell is basically a green technology. There is no emission, no toxic chemicals are involved over here, and no toxic environment is there because no heat generated, no extreme pH. So it is truly a green technology. Now, when this biofuel cell can be scaling it down, then we can get some more suitable characteristics which are for sensor applications. For example, the biofuel cell can be scaling it down to a very small size. Here I am showing it from our own example. We have reduced the size of the biofuel cell to a paper surface. So this, in the paper surface, we used one bacteria that is called cyanobacteria. The cyanobacteria can use as a catalyst in an anodic compartment, and surrounding is the cathodic compartment. So when you're scaling it down, it generates some new improved property. What are the improved property? One is that ohmic resistance is decreases. So as because it is smaller size, the gap between cathode and anode is decreases. The ohmic resistance is decreases. Sensitivity is increases. Response time is decreases. So these are the major advantages when biofuel cell is scaling down for its application, sensor application. So the other major advantage of biofuel cell for biosensing application is its stand-alone operation. Normally, most of the electrochemical devices which are used as a sensor, they need some external power source for applying the potential, whether it is a different kind of sensor, voltammetric or amperometric. But here, as because this device itself produces the current, so you need not have to apply the external power source. The current which is produced in the system will act as the signal. So this is a self-powered standalone operation. This is the major advantage of biofuel cell. Now let me describe briefly what are the applications of the biosensors in general, and then I will be describing what is the potential application area of biofuel cell-based biosensors. Now, one of the major area of biosensor application is the clinical applications. Now, historically, we know that the glucose detections, it has been developed in 1960s itself. So by one Professor Clark. So that is what he has developed. An oxygen electrode was developed, and later on it was translated to a biosensors by using glucose oxidizing enzyme on the systems. So since then, the glucose biosensors was the first biosensors, and since then many biosensors have been developed. But still, if we look to the biological samples, there are hundreds of analytes are there. Those analytes a few, reckonable numbers of analytes can be tested by using biosensors. Among them, glucose is one, urea, glucose, cholesterol, and few you can, recount. But there are so many analytes are there in the blood sample, in our stools, urines, saliva, and so and so. By measuring these analytes, doctors will tell you what is the diagnostic results. So accordingly, therapeutic interventions can be done. So for analysis these compounds, to analyze these compounds, generally what we do whenever we go to doctor generally prescribe and we go to the clinical centers. There we deliver our samples, and then we'll have to wait couple of hours and even days to get the results. But nowadays we have no time. We are also moving for in industrial countries, and people have no time. We need the results quickly. Whatever is going on in my body, I need to know immediately. So if those kind of biosensors are available, the way glucose is measured, then immediately, at the first hands itself, we will come to know if there is any aberration in our health, if there is any problem in our health. And then we can move forward. If we found that something wrong, then we can move forward to consult with a doctor. So we need a lot of those kind of biosensors. Now, not only for those analytes, you see the genetic disorders, genetic diseases. Since the completion of the Human Genome Project in 2003, it has opened up the avenue for vast sensor applications. Because after that genetic sequence is completed, then proteomics started, and we know the function of many genes. Since we know the function of many genes, mutations of those genes will also give you an information what will be the possible consequence of these mutations, what is the possible pathological conditions or aberrations that can be expected out of these mutations. So if you can precisely identify the mutations in the genes in your body by using some these kind of sensors, then that will, as a first, give you a hint whether your system is okay or not. If it is not okay, then you can consult with the doctor. Now it is not only clinicals, but there are enough scope of using these sensors for various applications. We know in Assam we are the major tea producers, but nowadays quality of the tea is tested mostly by sniffing. We know that tea sniffers are there, tea testers. So, but these are very much primitive kind of way of testing the quality of the tea. We need some precise sensors which can tell you the exact quality of the tea. So, but apart from human testers, there are some electronic nodes have also been developed, but electronic nodes have some problems because those are these are not very highly selective. So another area is that agriculture and food. There nowadays a lot of pesticides are sprayed, and then we purchase the vegetables to our home. But how we know that whether there is any pesticides or not? We cannot bring those cabbage and carrots to the clinical center to test whether pesticides is there or not. No, so we need some kind of sensors in our kitchen itself to check the level of pesticides, to check level of any food adulterations, to check level of any pathogens in our food itself. So that is very, very important for us. So that will avert any kind of disease. So there are a lot of applications are here, including the water quality. As you know, we have a lot of problem of water contamination with not only pathogenic microorganisms, but also various kind of toxic metals, metalloids like arsenic, mercury, and all these kind of things. If we have some kind of sensors for those, we can check those things in our kitchen itself to understand the water is potable water, is drinkable or not. So there are vast array of applications for biosensors. There is no end. Now, coming to the application of biofuel cell. The biofuel cell, as I have already discussed, that it is an energy producing. So it is yes, please. Anything? हम बैठा नहीं हूँ. So biofuel cell is an energy producing device. So one of the application for biofuel cell has been proposed powering that implantable devices. Now, in our body itself, we have enough scope for using this kind of implantable devices like pacemakers, blood pump, incontinence device, insulin pump, hearing device, neurostimulators, bone stimulators, and many more.
May I request everyone to kindly mute their microphones, please?
So I think that speakers should be muted. I request the auditors, please mute your speakers. Otherwise, sometimes I confuse whether I am asking any questions or what is going on. Okay, so there is enough scope for using these biofuel cells for implantable devices, because as you know that glucose can provide energy up to 16 kilowatt per gram of the glucose. We have sufficient oxygen in our blood also. It's about 200 micromole per liter. And because this oxygen will be required for cathodic reactions, glucose can be used in anodic reactions. So whatever the glucose and oxygen is present in our blood is sufficient to run a biofuel cell in our body itself. Now, this is no longer a hypothesis. It has already been proved. In 2007, one French group, they have already developed a biofuel cell and implanted in a rat, and that operates around 11 days with no ill effect. So it produces power about 24.4 microwatt per ml, which is suitable for pacemakers. So there is another area for application of biofuel cell, the wearable biosensors. You might have heard the Google Smart Contact Lens. This Google Smart Contact Lens is none other than a biofuel cell. This biofuel cell is implanted on the surface of the contact lens itself. And then the lacrimal fluid which is coming out when it is touched this contact lens, it produces the current, and that current signals can be transmitted. So different wireless technology like RFID, it produces the energy about 1 microwatt per centimeter square of the electrode surface at 0.5 volt. So that can be used to power sensors or other electronic devices. So this technology was actually running for a few years, but afterwards they have stopped it to refine it further. But this is a very good example of wearable biosensors using biofuel cell as a signal generating platform. Now, this wearable biosensors is not only in biomedical or clinical applications. This can be used for environmental monitoring also. For example, one Japanese scientist in 2016, they have developed a backpacked wireless sensors on the surface of the cockroach. That sensors can be implanted on the surface, on the backside of the cockroach, and then the sensors relies on the carbohydrate which is coming out from that cockroach backside, and that is enough for generating the power to run a sensor signal. It is, I think, a common phenomenon in our Indian household that cockroach is available. Most of the cases we are running behind the cockroach with our chapels, but ultimately we lose and cockroach win the game, because cockroach can enter in such a hole and such an area where we cannot enter. And there is a catch. That means cockroach can be present in such an environment where human cannot be present. So it can be present in some toxic environment also. If that is this, then this cockroach can be used to monitor any toxic gas on those environment. So that was the main concept. There is another area for wearable sensors that can be used as a tattoos on the surface of the body. As you know, this tattoo is one of the very fascinating things for young generations. So these biofuel cells can be produced in the form of a tattoo. So in this case, the fuel cells can be coupled in a series to generate more power. And then tattoo which is generated, it can be used for various purposes because it produces current, and that current can be coupled with different sensor systems. One such example is that sweat powered metabolic sensors. You see, in our sweat, lot of analytes are there, starting from glucose. The glucose level may be around zero point 0.1 to about 1 millimole glucose is present. So urea is present, uric acid is present, about 24 micromole per liter. Ammonia is present, various kind of salts are there. So there are many analytes are present in our sweat. So if biofuel cells will rely on those analytes as a fuel, then that can be used as a sweat powered metabolic sensors. You can use to detect lactate, glucose, urea, and many more things, ammonia. Because this will give you very critical information about the physiological condition of the body. For example, during that exercise, the lactate measurement is very important because lactate is produced as a result of that fermentation in the body in the skeletal muscles. So now the level of lactate will tell you the level of exercise you should do it. More lactate means more fermentative productions. So then the sports physiologist can suggest you what should be the level of exercise if that much of lactate is there. So apart from lactate, many things as I have already told you, you can measure pH, glucose, ammonia, urea, and so many things. So this is one of the good example. So this can be used as a battery-free biofuel-powered e-skin that harvests energy from human body, perform multiplex biosensing, and these informations can be wirelessly transmitted to a mobile user interface through Bluetooth. And in the mobile itself, you will come to know what is the level of different metabolites in our body. And then you can communicate with your doctor to central laboratory facilities, and you will get the feedback for your further course of action. Another very important area of the wearable sensor is for elderly care. You see, now our country is mostly young generations. As always, politicians told that we have a large number of youth, but these youth will one day they will be all be transformed to a senior citizen. Now we are also moving to an industrial countries. We have no time. We have to devote most of the times in our professions. Families are become nuclear families. So looking after parents is a very difficult situations. Now under these situations, if your parents are in your home, by dint of your this sensor devices, sensor area, you can monitor your parents or your dear whatever they are in home, you can monitor from your office itself. This is one example. In the body of a person can be implanted different wearable sensors. These will give, for example, various physiological signals can be derived like EEG, IOP, glucose, muscle movement, ECG, heartbeat, breath rate, and so many things. So sensors can be developed against all these kind of things, and that can be implanted on the body surface. And then once these sensors were implanted, these sensors can be coupled with your Bluetooth in your mobile phone or some those kind of portable devices. And then those informations can be transmitted to your emergency, to family or clinician, or if you are in your office, then you can receive those informations in your office itself. And then based on that, some interventions can be suggested. If it is an emergency, you can send that emergency doctor or vehicle to your home for taking care of your parents or the elderly one. So here, a lot of computational and not lot of modern electronic technologies have been used. Internet of Thing, as you know well, it is can work. So it works it can be worked through an embedded sensors. It involves the softwares, cloud systems. So basically, a cyber physical systems and an Internet of Things are now well developed. So those can be connected with the sensors to monitor a person from remote locations. So these are the some of the application areas of biofuel cell-based biosensors. Now, if you see the market of biosensors, it is a vast. A company, Market Research Private Limited, carried out one market survey, and they have reported compound annual growth rate about 8.3%. This studies was done globally, and they have found that particularly in America, they have a very acute problem, particularly chronic disease and lifestyle conditions such as heart disease, stroke, cancer, diabetes, obesity, arthritis, and many more. Now, monitoring these pathological conditions, repeat monitoring is a very important. But a country like us, or in the developing countries or developed countries, we have some additional disease burden. What are those disease burden? These are mainly infectious disease like tuberculosis, malaria, diarrhea, kalazar, and so many. Because these diseases are more dominant in underdeveloped poor countries or developing countries. So we need to develop sensors for those kind of things for our own applications. So there is enough scope of developing biosensors in those areas, apart from the biosensors for lifestyle diseases. Now coming to our main part of the discussion today, you see, we have discussed that electrochemical biosensors and biofuel cells. Both are electrochemical device, and both devices involves electrodes. Now the electrodes which are used are bioelectrodes. So bioelectrode means here we are using some kind of biorecognition element. It may be an enzyme, it may be a microorganisms, it may be a tissue, cell, or whatever it may be. Now the major question is that facile electron exchange between electrode and the reaction. So in these reactions, the electrons are generated. Our main objective is to channelize the electrons to generate the current, and this current can be used as a response, or the current can be used as a power source. Now, how this from the biological systems we can harness the currents to generate the response or to generate the power? That is the question. Now let us see our nature. What are the biological active materials we are talking about? We talk about various kind of microorganisms. It may be bacteria, it may be fungus, it may be some kind of algae, cyanobacteria. And as far as the enzymes are concerned, mostly we are talking about redox enzymes. That means enzymes which catalyze the oxidation or reduction reactions. Because electrons are generated, consumed or generated as a result of this redox process. So the enzymes which are involved in this redox process are only mainly primarily used for these devices. Now, if we look at the nature, the bacteria can produce current, or bacteria has an electrical effect. It is not a new thing. Actually, it was discovered in 1911 by one botanist, by one professor from Durham University, the Professor Potter. He was basically a botanist. So he first observed that when many organic compounds are decomposed, some electrical effects are generated. And these effects are coming from mainly microorganisms. Then after nearly 100 years, after nearly 100 years, because it was in 1911 and it was 2010, a group of scientists from University of Massachusetts Amherst, they have identified that bacteria can flush out electrons from their body. They have a mechanism. So these bacteria they have identified as Zeobacter metallodeusans. And this bacteria, this is the scanning electron microscopy image, the corner one. So here you might have seen some flagella-like wires. So these are basically not flagella. These are called nanowires. These are termed as nanowires. So this kind of bacteria can flush out electrons from their body through this kind of nanowires to the surrounding environment. And they have a beautiful mechanism for flushing out the electrons. What are the presence in the nanowire? It has also been studied, and they have found that there's a lot of heme clusters are there through which electrons are hooked from one cluster to the another cluster. Ultimately, it goes to the environment, surrounding environment. Now, why this surrounding environment? It has been hypothesized that many bacteria, which are basically present in the metal-containing ores, and if those metals are toxic to them, their first job is to neutralize the toxicity of the metals. So by passing out the electrons, they are reducing those metals, and thereby, to a certain extent, they are neutralizing their toxicity, and at the same time, those metals are also transformed to a soluble form, which can be taken back as a micronutrient. So these are basically the thought process. But what they have found that these bacteria can flush out the electrons to the surrounding environment, and this kind of bacteria are found in geobacter in a ore container. But later on, many studies have been carried out during the last 10 years, and it has been found that it is not only geobacter. There are many bacteria which can produce electrical current. So those bacteria are called electrogenic bacteria. Now, as we are discussing that bacteria are flushing out electrons, but if we look back our nature, then we can see that electrons are not only can flushing out to the surrounding environment. We have an electronic circuit in our system itself. One typical example is that we have that mitochondria. We have an oxidative phosphorylation chain. This oxidative phosphorylation chain, we have a lot of complexes are there, protein complexes are there, and through those protein complexes, the electrons are moved, and basically electromotive force is generated, and that electromotive force is basically an flow of electron. So we can term it as the electrical current. So electrical flow of electron, so basically an electrical circuit is already in our cellular system. So ultimately, the food which we are using are converted, particularly say when we use glucose. So the process of glycolysis and cyclic acid cycle, it is ultimately converted to NADH, and this NADH is ultimately oxidized at the first terminal of the phosphorylation chain, and then during that oxidation process, protons are generated, electrons are generated, protons from proton motive force, electrons from electromotive force, and then electron moves through the different complexes in a beautiful mechanisms, and then flow of electrons are generated. So this flow of electrons are already in our cellular system itself. So that means electrical circuit, we can say, is already present. That electrons, of course, ultimately at the end are captured by the molecular oxygen and converted to water. Now, this is not only in oxidative phosphorylation chain. In the plant kingdom, we have photosystems, PS1, PS2. There also similar electron flow mechanism is already there. But this photosystem is a little bit different from that oxidative phosphorylation. It is apparently it is the reverse of that oxidation phosphorylation, because in the oxidative phosphorylation, the water is formed at the end, whereas in the photosynthesis system, the water is split at the beginning, and from that system itself, the flow of electrons started. But anyway, what I would like to emphasize over here, that in our natural system, in our cellular system, there's flow of electron. The concept is already there. But interestingly, how this electron flows? The electron flows in a very beautiful mechanisms. The electron flows basically through hooks from one complex to the another complex in a potential gradient. And ultimately it helps the oxygen to reduce to water. Interestingly, this flow of electrons, rate of flow of electrons is exceedingly high. It is about 10 to the power 13 per second. So this speed of flow of electrons in a cellular system is amazing. Hardly it can be achieved by using man-made system. So there's a we can mimic the natural system. We can try to mimic the natural system. How this electron flow can be achieved to such an extent? It is so much of fast electron exchange. Now, well, I was talking about the flow of electrons only, but interestingly, if we again look back our oxidative phosphorylation chain, we see that natural biofuel cell system factory is already in our cellular system. If you see the mitochondria, if you see the mitochondria, which is always called the energy capsule, you see there what is happening in one terminal, whatever the food we are taking, glucose, etc., whatever it may be, ultimately in a citric acid cycle and a DS is formed, and the oxygen which we respire, oxygen which we take up from atmosphere, ultimately the oxygen is converted to water. How it is possible? So oxygen is reduced to water like biofuel cell. In a biofuel cell also in the cathode oxygen is reduced to water, and in the anode substrate is oxidized. And here also the flow of electron occurs, and the flow of electrons side by side the flow of proton is also occur. Main objective of this system, of course, the proton motive force, which is used to detach the ATP which is synthesized on the surface of ATP synthase. So main objective of our here is the proton motive force. But in case of that biofuel cell, what we are synthesizing, what is our objective, is the electron motive force, that means flow of electron, that means flow of current. So this is the basic difference. Probably we can learn something more from this nature, because here in natural system, the proton motive force is ultimately utilized, not the electron motive force. Electron motive force is generated to generate the proton motive force only. So in our fuel cell system also, if we can harvest some energy out of the proton motive force, which is occurred between anode and cathode, probably that will be another added advantage. Anyway, but let us see what is the mechanism, why we can say that it is a biofuel cell factory. You see in the anodic side, NADS is oxidized. The potential which is involved in the oxidation of anode is about minus 0.32 volt. Whether the oxygen reduction, the potential which is involved about plus 0.82 volt. So the balance potential plus 0.82 volt minus 0.32 volt is equal to plus 1.14 volt. That means this potential gradient which is there in a mitochondrial system, in oxidative phosphorylation chain, it is about plus 1.14 volt. So this potential gradient is created. Why? This potential gradient is created to create the flow of electron through different four complexes, as already I have discussed. So the four complexes, the electrons are moved from this terminal to that terminal, and then as a result of that, as because electron cannot flow alone, side by side in this biological system, the proton flow also occurs. The proton flow is ultimately utilized in another system, which will be revert back to the inner spaces for the ATP synthesis, where ATP is synthesized. I am not going detail about that. So we can say that this is exactly following the biofuel cell factor. Here in a cathode, oxygen is oxidized, in an anode, substrate is oxidized, and then current is generating, and the potential is produced about 1.14 volt. Now our objective is what? If we use microorganisms in a fuel cell, our main objective is to channelize the electrons which is coming near to oxygen to the electrode, debarring oxygen to take up that electron, so that we will get the current in a fuel cell. That is our main objective. So with this, now question is that our objective is to channelize the electron from there to anode electron. Particularly, this is anode. Now, can this electron be jumped from here to electron? What is the distance required for jumping that electron? Now, if we use enzymes on the electrode surface for developing sensors or biofuel cell, or if you use bacteria, whether it is a gram-positive bacteria or gram-negative bacteria, on the electrode surface to generate the power in the biofuel cell, the enzymes are very thick. You see, this hydrodynamic diameter of the enzyme is about 50 to 100 angstrom. So if the reaction will take place over here, can this electron jump to this distance? So much of distance, 15 to 100 angstroms. Or if you will use bacteria on the electrode surface to generate power in a microbial biofuel cell, can this electron jump about 40 to 60 nanometer, or about 35 to 40 nanometer in case of gram-negative bacteria. That is not possible. So what is the solution for them? We have seen that electronic circuit is inside this cell. We have seen that bacteria has electrogenic property. But the question is that how these electrons can be channelized to the electron. We have these challenges. Challenge is what? Challenges of the distance of electron hopping. Now, there are solutions. There are two solutions. Either of them can be utilized. One is that second generation bioelectrode we can develop, and second one is the third generation bioelectrode can be developed. Now let us talk about the second generation bioelectrode. In this process, we can use some small electron transfer mediator, which can shuttle the electron between the redox center of the enzyme and the electron. So in that way, whatever the reaction is taking place inside the redox center of the enzymes, the electrons can be channelized through the electron transfer mediator to the electrode surface. One typical example is that the glucose sensors. In fact, the glucose sensors which are available in our market, in a pharmacy or in our market or in the global market, most of these are based on this principle only. What they are doing the glucose oxidase as a biorecognition element to detect glucose, and in that process they are using one compound that is called ferrocin, or its derivatives are also used sometimes. So this ferrocin compound is immobilized along with the glucose oxidase on the surface of that electrode. And when you add glucose there, the glucose will be oxidized by glucose oxidase gluconolectone will form. And that electron exchange which will be taking place in the redox center of the enzymes will be siphoned through the ferrocin to the electrode, so then electrode will give the current. So electron will give the current, so that current will be the response. And through that response, the processor, we can see in a digital form how much glucose is there. So glucocord, glucorecord, or many glucose sensors are available in the market. Many of them are based on this principle. But these sensors are fine. But one major problem of these sensors is that the repetitive use of this sensor is difficult, because ferrocin which is there, it may not be interspaced for a long time. So operational stability of these biosensors may go down if you repeat, if you measure the same sensors again and again. So there is some thought that how to avoid this problem. So that's why the third generation bioelectrode come. There is no intermediate compounds are used between enzyme and electrode. But if you don't any compound inside these things, then what will be the mechanism through which you can channelize the electrons to the electron? So here is come that nanotechnology. Various kind of nanomaterials can be used, which can be used in the interface. So size of these nanomaterials is mostly comparable to the enzyme size. So as a result of that, due to their highly conductive nature, the electrons in the redox centers can change the presence of the nanomaterial, and that can channelize the electron to the electron. So there are various kind of nanomaterials are here. By using those nanomaterials, we can develop third generation bioelectrode. And there are many advantages of third generation bioelectrode is there, because if you don't use that mediator, those kind of sensors can be used in an open environment. Because most of the mediators which are used many of the mediators are toxic to the environment. So when no mediators will be there, you are using only the nanomaterials which are biocompatible and highly conductive, then those kind of toxic situations will no longer be there, toxic effect will no longer be there. And in addition to that, it has some many other advantages. So particularly these reactions will go hand by hand with the turnover number of the enzymes. And many other advantages. I am not going detail about these things. But most important thing is that the operational stability of the enzyme will be very, very high. It can be used again and again. But there are different kind of conductive nanomaterials are here. One is the gold nanoparticles. Graphene is currently emerging. It's one of the, graphene is occupying a huge share of market, particularly in sensor technology. And there are various kind of graphene materials are here. Graphene is basically a honeycomb structure, it's a highly conductive. Electron mobility is more than about 15,000 centimeters square per hole per second. So there are different forms of graphenes also there, like single wall carbon nanotube, multi-wall carbon nanotube, and so on. So these materials are helping the channelizing the electrons to imbibe the conductivity in the bioelectron. So apart from that nanomaterial, you see, we are also using biorecognition element for developing these biosensors or biofuel cell. Now, when we use these biorecognition elements, which are mostly enzymes or microorganisms, they are very labile. I mean, very sensitive to temperature, environment, and so kind of things. Now, these materials need to be stabilized on the electrode surface. Now, how to stabilize these materials on the electrode surface? We are using this nanomaterial fine. Nanomaterial will not affect these. This kind of nanomaterials may not affect the function of these enzymes or microorganisms. But at the same time, we'll have to take care their stability on the electrode surface for a long time. Because once you develop these sensors or biofuel cell, those need to be used at least for a couple of months. You'll have to keep it in a pharmacy for, shelf life should be high. So here comes some biomaterials. There are many emerging biomaterials. Among them, silk is one of the interesting biomaterials. It has been found that can be used to stabilize the various kind of enzymes that can be used to stabilize microorganisms, because silk can be converted to different forms. It can be converted to silk mat, silk films, silk hydrogels. So this way, it is not only is useful for biosensor development, but also it can be used for drug delivery and various kind of applications. And when organisms can be encapsulated with silk, that can be used for biosensors, biofuel cell, and even bioenergy sectors also. Now, I think I am coming to an end of my lectures. Just I am showing few of our products which we have developed. We have developed one biofuel cell using alcohol oxidase as biocatalyst and bilirubin oxidase as cathodic catalyst. So alcohol oxidase will oxidize the alcohol. Here we are using fuel. I think okay. The video I'm trying to okay. So this biofuel cell uh, we have demonstrated physically by using different loads. So stability of the biofuel cell also we have uh, validated. So this for this we have particularly used the device fabrication. For that we have filed patents, and the conductive being which is need to be developed for developing the anode and cathode. We have developed some special kind of graphite casting with silk sericin based materials. So that also we have filed the patents. And then recently this work also we have published in Applied Energy. Just in the last month it was published. So some of our products last year we have developed some products also, but not all are related to biosensors. One is formaldehyde detection, one is methanol detections, one is malaria detections. Malaria detection and methanol detections, of course, they are biosensors. So these are malaria detections of two types. One is pan-malaria detection, one is Plasmodium falciparum detections. So these two products, one is formaldehyde and methanol detections. Last year, Honorable Irrigation Minister Dr. Pokhriyal, he has dedicated these two products for the nation. Now we are working on these products, particularly methanol and formaldehyde, to include the gaseous formaldehyde and gaseous methanol also. And then malaria work also going on, particularly to give it finest level for technology transfer. So one just typical example how formaldehyde sensors we have developed. Just I like to display just 1.5 1 minute and 30-second video. Thank you for watching. So basically, now we are trying to accommodate gaseous formaldehyde also in this system, so that it will be a comprehensive formaldehyde testing kit, testing sensors. So similarly, we are also working on methanol sensors for gaseous, to accommodate the gaseous formaldehyde. So we are expecting in a maybe year or two to aspire this technology. So on this particular aspect, what we have discussed today, we have also one book edited, and that is available in the market. It also gives a hints about different kind of biosensors, emerging advanced materials for developing biosensors, smart materials, and different smart technologies which are used for developing biosensors have been incorporated here. So with this, I would like to conclude my talk. We got the support from Department of Biotechnology, Government of India. Actually, this support we are getting from 2008 onwards in different phases. First phase was, it was 2008 to 2013. With 4 faculty members, we are working on it with a total budget of 11.7 crores. Then second phase was given from 2016 to 2020. Then in that phase, we were given 7.75 crores. Then at the end of the second phase, DBT has identified few transnational product for our, and they have suggested us to submit another project. So accordingly, we have submitted. So they have given me 2 projects. So that we have recently started and will be completing in 2024. So we have these collaborations from Professor Melram from University of Alberta, Professor Estrella from University of Bath, Professor Eduardo Carton from Buenos Aires University, Devarstom from Irkutsk State University, Russia. With this, I would like to conclude my talk, and I request any questions. And I am thankful to all of you for patiently listening my talk. I'll be happy to address your questions. Thank you.
Thank you so much, Professor Goswami, for the very, very interesting presentation. And I'm sure all of us could get a idea of the level of research that is going on in the biosensors and biofuel cell lab in IIT Guwahati. So now we are open to a few questions from the participants. Anybody would like to ask something?